Hello and welcome back, erudites, to Next Level Catch-ups 2.0 for General Biology. In today's video lesson, we'll be discussing about Lesson 11, which tackles what we call as the overview of biomolecules. So let's dive straight into it with a short recap of what we discussed last week or in our last discussion. So in our last discussion, we discussed about, if I'm not mistaken, endocytosis and exocytosis, and how molecules enter and exit our cell membrane or pass through our cell membrane. So in today's video lecture, we'll be discussing about the molecules themselves, or are, what molecules those are, and also their different functions and their different subtypes. So let's dive right into the main topic at hand. So, what are biomolecules? So, biomolecules, if we break down the word, so bio meaning living and molecules meaning small things. So, biomolecules are essentially molecules that come from living organisms that have a important role in structure, functions, and also regulation of different tissues and organs. So it has four main types. We have the carbohydrates, the lipids, the carbohydrates, the lipids, the proteins, and also what we call as the nucleic acids. So um, these different four types have their wide range of functions and also have their wide range of subtypes. So before we get into that part of determining the four types of biomolecules and their different subtypes and different purposes, mag-ano muna tayo ng isang short recap. So, it's not a recap, it's more on a recall. So, um, in our previous years, we've already discussed about monomers and polymers. So I'm sure they've been discussed. So let's just have a quick refresher. So monomers, they are building blocks of a larger and complex repeating molecule. Meaning to say it's just a single brick sa isang brick wall. So yun na siya. Yun lang yun. Uh, to put it in layman's terms. So essentially, it's just a piece of a block of a piece. It's a piece of a bigger picture. So on the other hand, a polymer naman is several building blocks connected together to form a complex structure. So that's basically it for our short recall. So let's dive straight into our first type, um, first discussion. So um, in our lesson today, there are different polymers and monomers. So, as I mentioned earlier, polymers are conjoined monomers, and monomers are the most basic structures. So, how do we achieve polymers? So, polymers are achieved by conjoining two monomers, or in this first type of in this first process. So in this first process that we'll be tackling before we get into the main topic at hand is the dehydration synthesis. So the dehydration synthesis is a process wherein we take out water from a molecule, or rather we take out water from two molecules so that they'll be able to link together in a single, what we call this, in a single um, carbon link, or whatever link is available. So uh, these are made with simple sugars so that we can achieve a more complex structure. So on the other hand, a dehydration synthesis could also be reversed so that we achieve a more simpler structure, from a complex structure to a simpler structure, and that is what we call as hydrolysis. So before I tackle hydrolysis, this is the example I'm going to give you for dehydration synthesis. So in dehydration synthesis, we can see that we have a monosaccharide, two monosaccharides, which will form a um, polysaccharide. So in this example, we have glucose and fructose. So the glucose is this one and the fructose is this one. So as we can see, there are two oxygen molecules and two hydrogen molecules over here on the tail end. So we'll be taking a water molecule from both sides, or like in total, so that they can be joined by a single oxygen molecule. So what is a water molecule? Again, if we recall, a water molecule is simply two hydrogen molecules and an oxygen molecule. So we will be taking out two hydrogens and one oxygen. So that's this one already. We took out two hydrogens and an oxygen and left one oxygen to serve as a link for those two simple sugars. So eventually we got sucrose and water. So let's proceed to the next one. So hydrolysis. Now hydrolysis is a process that could reverse dehydration synthesis, or simply it could just make a complex sugar more simple. So how does this process works? So it's basically just the complete opposite of dehydration process of dehydration synthesis, where in dehydration we take out water. In this process, we add water to break down a bond. So in hydrolysis, we break down bonds by adding water. So by adding water, they'll be separated. The bonds will be separated. So why don't we see an example of how that actually works? So in this example, we did the opposite. We took a polysaccharide and made it into two monosaccharides. So we started with sucrose and we're supposed to end up with glucose. So in this example, you can see that there is water on the side, which we will add to the equation of the skeletal structure that is for sucrose. In sucrose, there is a single oxygen. There's a single oxygen molecule for it to be joined. So we'll be adding the H2O over there. So once we add H2O over there, we'll be having two oxygen molecules and also two hydrogen molecules. So they'll split evenly to form the glucose and the fructose. So that's basically it for hydrolysis. It just basically adds water so that it can equal out and balance each other out. So that's basically it for hydrolysis. So let's proceed to the main topic, the four major types of biomolecules. So the four major types of biomolecules, will start off with carbohydrates. So carbohydrates, they are carbons with water. So hydrate meaning water, and carbo meaning carbons. So carbohydrates are formed with oxygen and hydrogen and also carbon. So those are the basic ingredients for a sugar to be formed. So hydrogen ratio should always be a 2-to-1 ratio, and carbohydrates have different types of structures. There are more simpler structures and there are more complex structures. So essentially, it's a wide range over here. It's basically a cowboy's backyard field. So in carbohydrates, we have different types. First up, we'll be discussing about monosaccharides. Now, monosaccharides are simple sugars. These are simple sugars wherein it's called a simple sugar because it's the simplest form a sugar can have that we can know of or that we can achieve of. No matter how much we add water, we cannot achieve a more simpler form of this sugars. So the first one is the galactose, or the milk sugar. Second one is the fructose, or fruit sugar. Third is the ribose. Fourth is the deoxyribose, and fifth is the glucose. Now, just a quick note: the glucose is
for the blood sugar, but it's also one of the most common sugars that we'll ever find. And moreover, we'll actually see how it's one of the most common sugars. So let's move. So aside from monosaccharides, we have the disaccharides. So disaccharides are two monomers. Um, as you mentioned earlier, monomers are simple sugars. So aside from that, monomers in disaccharides, these are strictly only for two two monomers, meaning this is a complex structure, but it only has two monomers or two sugars, so that we can call it a disaccharide. So aside from the disaccharide, we also have what we call as the oligosaccharides. Oligosaccharides are more looser term but are still stricter, wherein it's 2 to 10 monomers to form a complex sugar. So there are sucrose, lactose, maltose, and raffinose. So let's dive deeper into those four types of oligosaccharides. In sucrose, these are made by fructose and glucose. So sucrose is one of those artificial sweeteners that we usually encounter in the various sweets or various foods that we usually consume. So next up we have lactose. Lactose is made up of galactose and glucose. So lactose is a complex sugar that, fun fact, humans can't actually. Uh, Humans do not originally have the enzymes to digest lactose. However, our body is adapted so that we could be able to um, we are able to digest lactose. Aside from that, we also have maltose. This is a glucose and glucose combination. It's found in different grains, such as wheat or something very grainy. Last up, we have ruffinose. So, ruffinose is one of the toughest because it's made out of three simple sugars to form a very complex structure. So, our bodies do not have um, the enzymes to process just two combinations. What more about three combinations? So it's one of those situations where it's very tough. Aside from that, our ruffinose is quite the type of sugar because it's also one of the sugars that aren't easily dissolved by any creatures. So that's that. So less up for the carbohydrates is what we call as the polysaccharides. So polysaccharides are a general term for anything that has more than one monomer. So a polysaccharide has many sugars. So first up is the starch. So the starch is a type of stored sugar from plants. So as we all know, plants produce their own food. So pag nag-produce sila ng own food nila, these come in the form of glucose. Now these glucose can't be stored forever in its glucose form. So therefore, the plants need to consume it unless they want it to turn into starch. The same applies for animals. However, a situation occurs wherein the stored sugar is actually in the liver. So once we consume food, the food we have to eat is the food that we can actually digest unless you want it to turn into glycogen. Glycogen. So um, in glycogen, these are the area where we store our polysaccharides, our stored glucose, the ones we did not digest because we ate too much. So these eventually turn into fats, which eventually is a long term source of energy. Last one is a thing that I'd like to put focus on, which is the cellulose. Now the cellulose is one of the most complex structures because it's a rigid structure that helps the plant maintain its shape and stability. So our bodies do not have the capacity to digest cellulose because we simply do not have the enzymes within the body, and we also do not have the amount of stomachs to be able to process cellulose properly. So cellulose can be processed by cows. And fun fact, cows have four stomachs, which have enzymes to process all of the cellulose in plants that they eat, in the grasses that they consume. As compared to humans, when we defecate, we can see that after we eat, there are bits of leaves. If we ate anything leafy, we can see that we there are floating leaves in the toilet bowl. So that's pretty much it for cellulose and its functions. So next up, we have the lipids, the second type of the biomolecules. Now, lipids are also called as fats, and they have structural supports they serve as structural supports, energy storage, or cell signaling. So let's dive deeper into the different types of lipids.
So for the different types of lipids, we'll start off what we call as the triglycerides. Now the triglycerides are one of the most um, common types of lipids that we observe or we know of. So triglycerides are a type of long-term source of energy. So as I mentioned earlier, glucose can't stay as glucose in our bodies, and same with plants. For us animals, specifically humans, our bodies can't um, can't can't let glucose stay as glucose in the body. So to solve that, our body makes the glucose and turns it into fat, into triglycerides. So if you really do not want to get fat, um, try only eating what your body can burn so that it won't be stored as fat. So aside from that, triglycerides are great insulators. Insulation because of the fat, and also they are great energy reserves. So this is a great energy reserve because it is a long-term source of energy. However, it's a long-term source of energy, but it also takes a long time to be able to take in the nutrients from that source of energy. Aside from that, we have what we call as for the next type, waxes. Now, waxes, we're very familiar with waxes. So waxes are very slippery. So uh, if you listen to a previous visual lesson, um, a great example of this would be in plants, wherein our plants have what we call as cuticles on their leaves. So the cuticles are a layer of wax on the leaves so that it prevents water from entering the pores within the leaves, but it also helps the water slide down the leaves to the root so that it would be able to get absorbed and be distributed properly. So, uh, another example of that would be bird feathers. So, why bird feathers? So, bird feathers are coated in a type of wax as well, wherein it prevents water from allowing the birds to become wet so that they can continue to fly. Um, they can continue be flying and their performance is not affected. So up next we have what we call as the steroids. Now steroids get a bad name for a bunch of reasons. However, steroids are actually a part of our bodies. They are a normal part of our bodies. So it just happens that steroids get a bad rap because of people abusing steroids. So, what are steroids? Steroids are there um, for different functions. They are widely for cell messen. They are widely used for cell messenger, uh, meaning to say they transport messages for our bodies. So aside from that, they have a wide range of functions. An example of steroids would be our hormones, our sex hormones. So now our sex hormones are designed to enlarge in some parts of our bodies, change aspects of our bodies, and that's basically what steroids does. And up next, we have what we call as the phospholipids. Now, phospholipids, we've already discussed this, but they are a neutral fat. Means say they do not favor water or oil at all. Rather, they are neutral because of its structure of being phosphorus and also lipids. So that's pretty much it for phospholipids. Up next is the third type of our biomolecules, the nucleic acid. Now, the nucleic acid has two types. We have the DNA and RNA. So, what are those two? So, the DNA, it's a double-stranded helical structure. So, it means it's a double helix, two strips. So, the DNA stands for deoxyribonucleic acid. So, and it stores genetic material and is double stranded. So, on the other hand, what does RNA do? So, RNA is the ribonucleic acid. It's a structure wherein it just copies the DNA. So, that is why it's usually a, not usually, it's always a single strand. So, we can identify it as RNA if it's a single strand. Um, so, let's dive a little bit deeper in nucleic acids with what we call as nucleotides. So what are nucleotides? Nucleotides are building blocks of nucleic acids. So they are formed by phosphates, this one over here, pentose sugars, and nitrogenous bases. So the in linking a nucleotide, in linking a nucleotide, the phosphate is attaches to a sugar, and the nitrogenous base attaches to a pair of to another nitrogenous base which is compatible with its nitrogenous base type. So what do I mean by that? Let's look to the next slide. In this one, um, as we already know, there are different types of nitrogenous bases. And these nitrogenous bases, we can see that there are different pairings depending on what type of nitrogenous base. So for adenine and thymine, they are pairs for duplication. Cytosine and guanine, they are pairs for duplication and replication. And then adenine and or adenine and uracil are both pairs for DNA replication, so RNA. So that's primarily it for nitrogenous base pairings. So let's discuss the last part of this lesson, the proteins. Now, ano ba yung proteins? The proteins are polymers of amino acids that are joined by peptide bonds. So, what are amino acids? Amino acids are the monomers of proteins. So, they are the building blocks of proteins. Aside from that, proteins have two helical structures. They are called the alpha helix or the alpha form, or and also the beta helix or beta folds. So let's look at that one. So the alpha helical structure and the beta folds. Oh, I'm sorry. We'll be discussing about peptide bonds first. So in this instance, I'll show you what I meant about peptide bonds and peptide chains. So this one is a nitrogen is a amino acid. These little things are amino acids. The connecting part between amino acids and amino acids is what we call as the um, peptide bond. Once there are three amino acids bounded together, that's a peptide chain. Once a peptide chain has formed something else that is bigger, that is the only time that we can call it a um, that is the only time that we can call it a polypeptide chain. So polypeptide meaning many peptide chains are present within that link. So that's it for the peptides of proteins. Let's move on to the folds and helixes of a protein. Now, as we mentioned earlier, um, proteins are links of amino acids. So our amino acids link together to form helixes. Once they've formed helixes, they'll eventually have to fold themselves and figure things out to form beta sheets. So that's how it goes from a amino acid, then a peptide chain, then a polypeptide chain, then a helix, then B not a alpha helix and the beta sheets. So that's primarily it for the structure of proteins. So aside from that, we have the different types of proteins. So first up, we have the structural proteins. Structural proteins are types of um, proteins that help our bodies maintain its shape. So example of just of this would be collagen, wherein the I believe our joints are maintained by collagen so that it stays strong and healthy. Next up, we have the elastin. On the other hand, elastin makes our body flexible. So, still structural because it provides structural support by allowing us to be more flexible. But yeah, that's primarily it. Up next is what we call as the defense a function or type of protein. In the defense function, it's primarily there to observe if, pardon me, if there are foreign bodies entering the body. So an example of this is the immunoglobulin. So immunoglobulin are important in maintaining the purity of blood. It's there to safeguard blood and also act as a defense system. Next up, we have the transportation type of proteins. So an example of this would be hemoglobin. So our hemoglobin carries the oxygen that we got from our lungs into our bloodstream and to wherever it needs to go. So it's very important that we have hemoglobin, specifically transportation protein types, so that we'd be able to transport nutrients. So yeah. So aside from that, we have um, the secretory type of the proteins. Now, the secretory type is a type of protein that's main goal is to speed up the process of things. So an example of this would be our not sexual, reproductive hormones, the testosterone, progesterone, and the um, estrogen. So these hormones, they enlarge certain parts of our bodies. They hasten or speed up different processes, such as just to name a few, widening our hips, broadening our shoulders, making the vocal folds of our bodies to be thicker so that our um, voice would be lower, and so on. So aside from that, we have the storage function. The storage function is very not discuss a what do you call this? Storage function is um, self-explanatory. Let's move on to the movement type. So, what's the difference between the movement type and the transportation type? The transportation transports molecules from the outside to the inside. Now, the movement function
is actually the movement of our body itself. So, an example of this would be actin fibers. In a previous discussion, we discussed that actin fibers are fibers within our body that allow our bodies to contract, to stretch, and so on. So that's basically it for the movement types. Last one is the receptor type of proteins. So the receptor types of proteins are important in dealing with our immune system. So these receptors stimulate a response so that we could act appropriately, or our bodies should act appropriately to depending a situation, whether we're sick or not or something else. So that's primarily it for our lesson tonight, or well, rather today. I'm filming this at night. I'll catch you all next time, erudites. See you and thank you.